and we could see immediately those tubes. They are connecting them each to, and we later found they are membranous in nature. It's not pillin or pili or like cytoskeletal molecule. Could see we observed it by electron microscope in Zurich. The nanotube established a network of tubes that provide a route for exchanging cells. Whether indeed nanotubes provide a path for that, it's similar experiment. We took FP producing and non-producing cells, fused them together, and then observed by immunoelectron. And you could see clearly those FP molecules coming from. So that establishes indeed the. So whether this tube formation between the cells can be beneficial together or it's just so. For that we had assumption that one will take just one kind of antibiotic resistance bacteria and the another kind of antibiotic. And if we grow them together, idea was there would be formation antibiotic resistance. That says whether this is exchange or if plasmid as well, hereditary, non-hereditary. Because if there is protein exchange, non-hereditary, plasmid. For that, spotted wild type, one strain kind of antibiotic. Mixed, there you could see that when one kind of antibiotic, that chlorum can grow at one place and mix can grow. Others one cannot grow here. I mean, they can grow here, but not here. That proves okay. And only goes on both antibiotics. That proves for that whether it is just or what, that we took that grown plate, this one, going on, and then we done simple LV, and then we spotted on LV this single plate, this one, only those two plates did grow. That means there is change in the molecule. Clearly did that acquiring hereditary. For that we took another mix which is harboring plasmid, and similar experiment only when we spotted them on triply resistant plate, only these strain can grow, this not. Here we clearly observe that okay, there is a protein exchange which is non-hereditary and there is a plasmid exchange. So what is the molecular composition of these two? For that, like what are genes that are involved in? Then we did a lot of screening, knockout, clean knockout, and then we found, at that time we found YMDB as a molecule that is responsible. for the exchange. See, if you knock out this, no strains, they are like mixed strains not growing out any plate. So whether this YMDB protein, whether this is really responsible or it is localized to the nanotube or not. So we did a single molecule analysis. This is palm, and we just labeled YMDB with GST, like tagged YMDB gene with GST, and we observed them by using correlative microscopy as well as non-coding scanning electron microscopy using GFP as a antibody, and then we did correlative. Here you see that uh, the green molecules, they are perfectly correlated the nanotube. So that means we proved it here. Okay, this molecule, this is responsible for the nanotube formation. So what is the consequence of having these nanotubes, physiological consequence? For that, when we took wild type, they grow overnight. But when we do knockout null YMDB, they cannot grow. I mean, they have a small colony morphology. Then next question was, okay, let's complement it. Then we put YMDB back in the genome, and then you can see the similar complementary strain and wild type, they show that. Then there was a question whether we can follow them from the almost relatively low cell density. That we took the strain, we labeled YMD, followed it. And there was one more observation that we found it. When bacteria grow, they define their territory. Like they define as to how, like it's like our colony. It's like a neighborhood. So they define the territory and they grow centrally and they have it. And because when we plate the bacteria, why we always question why they grow only like circular at this distance, why they cannot spread all over to the plate. So they have their limited territory, and that says bacteria, they have arms, especially Bacillus subtilis. They define their territory and then they grow centrally. When we knocked them out, you can see that they don't grow clearly. Like they don't define their territory. So that says, okay, there is multicellular behavior in bacteria which is mediated by this molecule and nanotubes. So in summary, nanotubes run a route for cell-to-cell communication. Nanotubes mediate exchange of cytoplasmic molecule. I mean, we later found, okay, this can also mediate membranous molecule, but I mean, we did not publish, including resistant antibiotics when they live together. So, nature nanotubes play a role molecular exchange and emergence of antimicrobial resistance, MR, in bacteria. That was when I started lab in last April, but main work was done with Sigal Ben Yehuda and Philip Cincinnati. That I'll present with Philip Cincinnati. I'll present maybe later, or in my proposal I'll show what is the main project where I fit to AIG. Can I have my um, There is one more file. Yeah. Maybe. You can interrupt. Anybody can interrupt. Yeah, please. It's a normal discussion. Please go ahead. No, we first found nanotubes. I mean, people have seen that, but I mean, it requires a kind of very cutting edge microscopy.
People have seen it, but it is not.
It was not questioned whether why it is happening.
Oh.
People have seen that. I mean, I got a letter from Germany, like 70, 80, a scientist, and he said, okay, I mean, we have seen that, but we like imagined.
Importance of the thing was never known.
Importance they did not understand. Yeah, because they just believed, okay, this is quorum sensing and all membranous vesicle and all conjugation. But no, it was all independent of conjugation. The strain that I used has no conjugative apparatus. So they cannot make conjugative pili. I mean, we also did that first. So they don't make a conjugation system. Yeah. In 2011 and 2016. Their cover page. I mean, they covered me all over European media and all American media, everywhere. Yeah. So for so yeah, your question is like that. For that we did this we wanted to found because people ask the question, what are the genetic material that is responsible for the finding? First thing, and second thing, if I understood correctly, is like donor to recipient who makes, but it's both way. Like
Random occurrence, or is there some trigger which makes it this two-way process?
so,
Yeah.
the question. So when they live together, bacteria. So like Lederberg, what he did in while asking the conjugation. So he took kind of stationary 20 rpm. So he took that, and where he observed, took LB plate and then spotted them together, and then they grow, like they make the nanotubes, and we don't know what are the physiological conditions yet. We don't know. No,
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 it's
no.
just co-culturing,
Yeah, co-culturing together.
no
No, no.
antibiotics.
Only on LB plate. No.
And can you manipulate that to stop this? Then it makes clinical translational sense. I mean, through a mechanism now, there is exchange of genetic material for resistance, and that's how they propagate.
Right.
But is there any way to stop that? That nanotube, can it be stopped, or the transmission of the genomic material can
Yeah,
be stopped,
so,
so that
for-
resistance can be avoided?
For that, we found because this is a kind of like existing in nature. So we found that okay, now our idea is, so for that we found YMDB. Now our idea is as to how it happens in the environmental strain or isolates like salmonella, this and that. If we co-culture them together, and can we stop them or not? And whether really only YMDB is responsible, but it's not true only a YMDB is there. No, it's not one gene. Now they found later on, my juniors like postdocs, they found there is a core like flagellum. There is a core, and here the strain that we used knockout flagella. Now they found that there is a kind of flagella like structure, what they say, a nanocore. So they found a lot of genes which are responsible. So it's like kind of a system. It's not only one gene. It's not just like a pillin or pili or something.
Why you just asking? I mean, just extrapolating it, we feel that cancer cells multiply. Now I don't know whether you're talking about this nanotubes connecting cells to cell, whether cells are multiplying or one cell is going on to the spreading from one cell to the other cell.
Yeah. So in mammalian system, so they have this what they call mammalian nanotube, and in cancerous cell they also found, and they said they have proved that they promote the virus transfer, all these mitochondrial transfer and all these, they feed it them together.
Is there any particular genetic material that is transferred or is it random? Because if you are doing the metagenomics and if there is transfer of genetic material, are we classifying the bacteria wrongly based on the 16S classification? Do you think we need to look at the bacteria itself?
No, I could not get. Could you just simplify that question?
Usually when we do the metagenomics, we isolate the DNA and look at the specific region in the bacterial genome to classify the bacteria. Now, if there is exchange of genetic material between bacteria, are we classifying the bacteria wrongly because there is exchange of genetic material?
Oh, that is yet to be answered. We haven't done metagenomics. We did clean knockout, transposon and CRISPR and all this stuff. We did not do the metagenomics. That's what we are targeting now, which is yet to be undisclosed. That we will take environmental isolates and all, and then we are also, I mean, people have found that these nanotubes are in gut microbiome. Like they exchange uh, uh, between gram negative to gram positive, and we had already cleared that okay, this is interspecies man, it's not only between one or two bacteria.
That was his question. Suppose you make a conjugation tube or nanotube between a gram positive and negative, and there is transfer of material. Does the gram
No, it's
positive
not
become gram negative?
no. So we used bacillus as a model, and then we used stuff.
Your thing is okay. You are using only one strain, but his question is in general. If you take the whole picture,
That
you said gut microbiome. It has been shown that there is a nanotube between gram positive and gram negative.
in the yes, gut
Yeah. So
microbiome.
if there is a transfer of some material from gram negative to gram
So,
positive,
yeah, so
does the gram positive become gram negative now?
So
Because
it's-
there is the 16S RNA comes there.
So still, this question has to be answered because people took one kind of bacteria which is gram positive and another kind of bacteria which is gram negative, and they make nutrient feeders. Like one amino acid, one bacteria expresses another amino acid, another bacteria, but they cannot live because they are auxotrophic for each particular amino acid. But when they co-cultured, that paper was from Czech Republic or so. So when they co-culture, they grow. So it's not specific. Only the point here is when they live together, and they are membranous, so they can fuse. Still, people ask question that how they cross, like gram positive or gram negative bacteria, they cross peptidoglycan. So that's the one of the key question yet to be answered, and Sigal is working on that. Largest. So it goes like around uh, what we measured, quantified it, that it can travel. So there are two things. One, so we found two kind of nanotubes. When they live together, they make the nanotubes, and they are smaller, like around 3 to 4 micron. But when single bacteria they live, they have, so this is intercellular nanotube when they live together. When the single or relatively low cell density bacteria, they are growing, so they make scavenging tubes, and that scavenging tubes we proposed, we could not prove it. We proposed, okay, they are traveling for nutrients or they are traveling for some kind of molecules or trying to feel physiological conditions or what. We couldn't prove. it's like eukaryotic cell, they take dextron and they uptake it and all. We tried it, but it's hard. They're very tiny.
But if this works out, then there could be lots of translational impact. If you can change the pathogenic gut microbiome by nanotube and putting in genetic material, the pathogens can become common cells.
Yeah,
So it's a
that's
concept. I mean,
yeah. That's the main point. Yeah. So that's you're talking about gut dysbiosis and all. If they are living together, okay, yes, a commensal can become pathogenic, and pathogenic they can. I mean, that's how we establish commensalism and dysbiosis, leaky gut or what.
Do you think nanotube with the host cell in case they are in the human?
In human, yeah. So yeah.
Exchange of
So
genetic
follow up later said that in EPEC system, type 3 system, secretion system. So these bacterial cell, EPEC cells, they mediate they form like they inject type 3 system through the nanotube. That was also again published in the cell, but it was follow up work. Yes, they do make it, but still it's like preliminary, more to be explored, and very intriguing and all.
Another thing we thought that electron microscopy is basically a technical tool, but does it have a functional component also wherein you assess the functional also? Electron microscopy, where you're talking about the function of this thing.
put some gold particles and see there is shift in gold particles after treatment. So it can give you indirect
There's some
evidence of
functional
function.
component
Yeah,
also.
indirect evidence you can get. You have to use like specific immunogold techniques or some specific.
But what sir asked that okay, we have to analyze and use that model system with environmental isolate and get gut microbiome and just to see and do the
Maybe your
functional
next
16
step should be
sequencing.
rather than using only Bacillus subtilis, you can use different like multi-strain or different species altogether and see how they crosstalk to each other. That
Yeah.
will have more of a translational
Yeah.
value.
That's the
Yeah, I
next,
mean, this is the first step, I understand.
yeah,
but,
that's the next step. I mean, that's one of my project goal. Yes. Yeah. So I just want that my research proposal. That, sir, can you help me out to, because I want to get into the gut microbiome a bit. That scientific proposal. Scientific. No, no one. I just want to. So, like the title I said, yeah. So we have the like background of this thing, bacterial nanotubes, a new strategy, a novel phenotype that how they originate, and then gut bacteria, intestinal cell interact. And we have a few questions up there. In one, how bacterial multicellularity originates. One, now I'm how multicellularity originates, and again to get to the environmental strengths, whether nanotubes mediate these communication. how do commensal gut bacteria and intestinal epithelial cells, basically targeting to the ileum, how they communicate and how there is a dysbiosis and all in. So on second question, we went to the. So for that we used so yeah, here it. So here in second figure, figure 3, you can see we used live cell imaging and we immediately fixed them like in Titan, the T300 cryo-electron microscopy, and then we did tomography. It was even hard to get because they are 70 nanometer thick, so it's hard to get to go through it. So for the gut microbiome system, what we used a model system segmented filamentous bacteria. Uh, so and they modulate all the immune system, almost all the immune system. And then we took the germ-free mice and notobiotic mice. Like we took the germ-free mice, cultured them like ground 3 and all, and then we gavaged with this specific bacteria, oral gavaging. So that we made notobiotic mice. We got it from Japan and all. But then we developed this system. So, but it's very hard for the gut bacteria because they are so anaerobic and so auxotrophic. Because this bacteria they had like this one SFB had almost 3.8 or something, very short genome, and it was all dependent on the nutrients to gain from the host cell when they live together with the hosts. Highly auxotrophic. So, first paper that was published by my senior, that he tried to mimic the uh, host in vivo system, how the bacteria use different media and all, and then she could manage to co-culture this bacteria with the intestinal cell, like TC7, Kapo's cell, MICCL2 and all. So that was published in Nature as well, because it was the first time someone demonstrated that gut bacteria can be co-cultured with the intestinal cell. For that she used all hypoxic system, low oxygen and all and all. And then we later wanted to understand what are the apparatus that could mediate, because mine is more of this, the apparatus, because I had to all these uh, microscopy and all these observations. So then she left, she got the job, and then I followed it. For that, what we did here is we did fish cultured here long, very very long. You can see by naked eye, like they are like a centimeter long. They grow like this. When they go to single, only this one, they have this mediates their interaction, and then they grow long, long, long. They make sports, indoor sports, and all the office that we, and then they start their cycle, and then they whole life and regulate the uh, post immune system, all the system. And this is very fascinating that it, I mean, still it's debatable that if they are present in human, but some reports already said from Italy and Czech Republic that they are in humans. So one of the main target here is, I mean, I have probes and all to take infants of 3 to 11 years along with the mouse model, both male and female, and to do observe like 3 weeks, 4 weeks, or 3 years, 4 years, to observe whether they are present really or not, and what kind of species they are, and how they regulate. So this is one of the key objectives. And here is a design that we will do that what I said is male and female. First we will try using mice model, and then here we will get sample, I hope so, that then we can try to observe in infants both male and female until 11 years, or we will follow up until next like other ages. Yeah.
So how did you maintain the notobiotic mice?
In germ-free facility.
But here, I mean,
Yeah,
India,
that's-
India, I don't think in Hyderabad, 1 or 2 centers was like prohibitively expensive.
Yeah,
Half of the
I mean,
research
it's very
goes there.
expensive. Yeah.
Half of the
I mean,
research
in
goes
India,
in
like
maintenance.
what Rajesh Gokhale said, now you have time to propose, okay, you want to have a first notobiotic system in India, but he said no, for that you have to recruit another 3, 4, you have to build underground and all this. So that's what I'm limited, but still I'm in touch in Japan, Riken, and in Pasteur Institute, other animal facility. I can do there, but in India, I mean, it's hard. Yeah.
Notobiotic is highly prone to infections.
Yeah, yeah.
Whatever level of biosecurity or sophistication you have
So
in the
we go
facility.
like so in Pasteur Institute, Pasteur, they were like underground 3. So there is no noise, no infection, nobody can go, only trained people can go, and they are very prone. If they get noise, okay, so there is no, like, but they lose the bacteria. So we have, I mean, we used to get infection, infection very often, other bacteria, but it's hard and it's very rare to get a germ-free system. Now in China they have, at that time it was in Pasteur, Germany, and somewhere in, I mean, in USA now they have a lot, like a Jackson laboratory and all and all. Now they have it. So yeah, it's difficult. In India, it's hard to propose this. But what I am thinking is to observe using model system, mice model system, animal system, and also especially in cattle and all. So that is one of the objective. I mean, I'm not proposing to have germ-free facility at the moment. I mean that if you understand its credibility, yes, we can do that. It's not. It will take. I mean, we can propose it. At this moment, if I will ask, they will ask me, you are asking for the job or the facility or something else. Complicated.
That be the study will be for 3 to 11 years. You want to track the animals for 3 to 11 years.
Yeah.
In
Because
mice.
in mice and in like humans, because they disappear.
Yeah, see, mice is maximum 2.5 years lifespan. So you propose to take the microbiome of the mice and then transfer to some higher animal, or what do you want to?
We want to observe as to how their colonization happens over time, and then we want to go back to the humans. as to how they correlate. That's one of the things. We have mice from Hylesco and trying to do that. Yeah.
One question I had, you said this particular nanotube appears in the stationary phase
No, no,
or log phase
uh, uh, no exponent in the stationary phase they disappear.
throughout.
Oh yeah. So from the exponential phase, they grow. In the stationary phase, what the model system I used, in stationary phase they make spore. So no, I mean we didn't. I mean we could not observe, or rarely we observed. The key point here is you have to have low shaking culture like 25 rpm or some solid media, and then allow them to live together so they can establish.
So does
If you
the-
take the shaking culture to 250 rpm and all, no, they don't make.
does this nanotube formation have effect on doubling time of the bacteria?
No, no. I mean,
See,
we did not go into that question, doubling time, because Bacillus had doubling time of almost 30 minutes, Coli had doubling time of almost 20 minutes. So, I mean, we didn't question, I mean, that time, but what you ask, stationary phase, in stationary phase, no, they don't.
so do you think there's a trigger, probably nutrient depletion is a trigger for this formation of nanotube?
Yes, nutrient yeah, of course. Like antibiotic deficiency, yes, they do allow. Nutrient deficiency, yes, they do make, because what I said, if you take two bacteria which is auxotrophic to one strain, another one amino acid, another bacteria, another amino acid, they, I mean, they cannot grow alone, but once they make, they trigger it. They can grow only when they live together. Like if you take alanine lysine, so if one is auxotrophic to one strain, so they cannot grow to alanine. If another one is auxotrophic to lysine, they can't grow. And when they live together, they make. But what you are pointing out, physiological conditions, I mean, we could not, I mean, at, at this time, we could not decipher. Backside, come on, come up.
Thank you, Dr. Dubey. There is no questions, so we're going to close this.
Come up. You're not curious. Basically.
Maybe
Oh yeah, please come up. Yeah, yeah. Why? That's so. That's why I explained. If you knock out IMDb, they don't make. They don't define their territory. They just get a small colony. So if you overexpress IMDb, and then they define the colony, and then they.
Want to tackle this problem of antimicrobial resistance. So what approach do you think would be ideal? Because if we are trying to knock out YMDA as one of the approaches, then we are actually hampering the entire growth process itself. Like the colony formation itself is getting affected.
Right. Yeah.
So did you find any genes which are actually facilitating the transfer of the genetic material from one bacteria to the other bacteria? Because if it is transfer of like proteins, there would be some elements which is facilitating it, and for genetic material it would be something else, like maybe identifying that factor which is facilitating.
this is question to be answered. Like follow-up studied this are saying, okay, this could be specific to the uh, protein transfer. This could be specific to the plasmid, like non-hereditary and hereditary. And my next target is to take the uh, antibacterial resistance, like I take the isolate from different places and then co-culture together, and just to ask and do screeners as to how and what mediates. It's
Yeah,
just
I'm talking about the signal, the cue, which is actually telling that this bacteria needs DNA transfer, like hereditary transfer, and not just a non-hereditary. So maybe identifying that signal would help us in tackling
yeah,
this problem of AMR.
that's one of the question to be answered. Yeah, we could not decipher. Yeah, that's one of the question that has to be answered. What are the signals that trigger
Yeah.
and all the stuff? Go ahead, come up. Go ahead, don't be hesitant. So there are no queries. I thank you all being here on your time.